Good morning, students. Hope you be fine at your respective places. In the last chapter, in the last videos, we were discussing the different types of the RNA. We have discussed the mRNA. We have discussed this tRNA, and today we are going to discuss third type, that is the rRNA, or it is also called as the ribosomal RNA. Ribosomal RNA, or in short it is written as rRNA. And this rRNA, अगर हम इसकी बात कर लेंगे, it is found in the, why it is called as the rRNA, because it is found in the ribosomes. They are found in the ribosomes. Now how we can prove this? अब हमको ये कैसे पता चल जाएगा that they are found in the ribosomes? Dear students, you know that अगर हम राइबोसोम्स की बात कर लेंगे, राइबोसोम्स दैट इज इक्वल टू rRNA प्लस प्रोटीन. So this ribosome, it is rRNA plus protein, and this, ये हमको इससे हम पता चल जाता है कि that these rRNA, they are found in the ribosome, and this rRNA, it forms about 60% of the ribosomes, and this protein, it forms about 40% of the ribosomes. Now एक और फीचर की अगर हम इसकी बात कर लेंगे, this rRNA, it is insoluble RNA. अगर हमने बात की tRNA की, tRNA में हमने कहा that it is also called as the soluble RNA because it gets dissolved in the 1 molar NaCl solution. Now this is the insoluble RNA, as it does not get dissolved in the 1 molar sodium NaCl solution. This is second. थर्ड अगर हम बात कर लेंगे इसकी, which I have told you that it is found in the ribosomes. These ribosomes, they are the sites for these photosynthesis, this protein synthesis, and this ribosome, it is made up of subunits. Also, अगर हम बात कर लेंगे, अगर हम बात कर लेंगे राइबोसोम्स की, सी है, अगर हम बात कर लेंगे राइबोसोम्स की, we have got mainly two types of ribosomes. First one that is called as 70S ribosome, and this is the second type, it is called as 80S ribosome. This 70S ribosome की अगर हम बात कर लेंगे, it is mainly found in prokaryotes, while as this 80S ribosomes, it is mainly found in eukaryotes. The eukaryotic cell, it is found in prokaryotic cell. Now we know these ribosomes, they are made up of subunits. Now this 70S ribosome की अगर हम बात कर लेंगे, it is also made up of two subunits. First one, 50S subunit and 50S subunit and 30S subunit, while as this 80S ribosome की अगर हम बात करेंगे, it is also made up of two subunits, that is 60S ribosome and 40S ribosome. Now this rRNA, it has got different types. It has got different types. Now when we see what are the various types, this 50S अगर हम बात करेंगे, this 50S ribosome की अगर हम बात करेंगे, 50S ribosome की, it has got two different types of rRNA. First one, that is called as 5S rRNA, and second one is 23S rRNA, and 30S की अगर हम बात कर लेंगे, it is made up of 16S rRNA. Now this means that the 70S जो हमारे पास है राइबोसोम, it has got 3 types of RNA. In the bigger subunit we will see it has got 5S and in this and 23S, and in the smaller subunit it has got the 16S RNA. Similarly, अगर हम इस 80S eukaryotes में देखेंगे, here we will see 3 different types of rRNA. First one, we will see here it is 5S rRNA, 5.8S rRNA, and third जो हमारे पास है that is 28S rRNA, and these are the 3 which are found in this 60. These are the types of rRNA, and this 40S rRNA की 40S जो smallest subunit है, it got 18S rRNA. 18S rRNA. So when we see in the 70S we got 3 different types of rRNA. In 80S इसमें हम क्या देखते हैं, यूकैरियोट्स में it has got 1 2 3 4 different types of these rRNA. So it suggested that this rRNA they are also found in the this ribosomes. Now अब क्वेश्चन अराइज हो जाता है कि ये कहां पर बनता है. Where is its place of formation, where it is synthesized rather, where it is synthesized. This rRNA, अगर हम इसकी बात करेंगे rRNA की, it is rRNA, it is synthesized in nucleolus region या nucleolus region of the cell, वहां पर ये पाया जाता है of the eukaryotic cell, वहां पर ये पाया जाता है. Now अगर आप कोई भी यूकैरियोटिक सेल देखेंगे, वहां पर हम देखेंगे क्रोमोसोम देखेंगे. क्रोमोसोम में हमारे पास एक रीजन है दैट इज कॉल्ड एज NOR, and that simply means nucleolar organizer region. It is that region where we will see this rRNA is synthesized or it is formed. So when we see the eukaryotic cell की अगर हम देखेंगे, वहां पर हमको क्रोमोसोम देखेगा, that is called as the eukaryotic chromosome, and it has got the region which is called as NOR, which is the nucleolar organizer region, where this what we will see this rRNA is going to be synthesized. एक और बात जो हम यहां पर इसके मुतालिक कहेंगे, we have got different types of the rRNA, लेकिन and these are different types of RNA, they are present in the rRNA. About ये देखा गया है 80% हम देखते हैं कि rRNA is found in RNA. About 80% of rRNA, ribosomal RNA, is found in a RNA. Again, we will see one more 10 to 15% of 10 to 15% of RNA, या हम कहेंगे 10 to 15% of tRNA is found in RNA, तीसरी बात अगर हम बात कर लेंगे, दैट इज दिस 5 to 10% ऑफ mRNA इज फाउंड इन RNA. तो इससे हमको पता चल जाता है कि RNA में ज्यादातर जो परसेंटेज है दैट इज दिस rRNA या राइबोसोमल RNA की बात है. फिर 10 to 50% ट्रांसफर RNA का, और 5 to 10% mRNA दैट इज द मैसेंजर RNA. एक और बात अगर हम कहेंगे अबाउट दिस RNA की, दैट दिस RNA it is the most suitable form of किसका, most stable form of RNA. This rRNA, it is the most stable form of rRNA. Next, अगर हम इसकी एक और characteristics बताएंगे, what is that we have got an enzyme that is called as RNA polymerase first. We have got these three: RNA polymerase second, RNA polymerase third. I will give you the detail. Help us in this. It helps us in the synthesis of. It helps us in the synthesis of rRNA. It helps us in the synthesis of rRNA, or we can simply call as ribosomal RNA is synthesized by with the help of this enzyme RNA polymerase first. Now we have got two more these polymerase first. We have got RNA polymerase second, yeah, RNA polymerase this third. This RNA polymerase second, it helps us in the synthesis of mRNA, and third जो है, it helps us in the synthesis of tRNA. And this RNA polymerase first, it helps us in the synthesis of RNA. Now, एक और बात regarding this RNA की, if we talk about this, that this RNA, it is weight जो अगर हम बात करेंगे, of the three different types of the RNA, it is the heaviest. It is the heaviest RNA. Now, let us try to, this was, it is what we call as the structure about this RNA. Now, let us try to describe the functions of this rRNA. Functions. It performs some important functions, and the first one function which is being performed by this rRNA, that is it catalyzes, that is it enhances, it increases, it catalyzes protein synthesis, that is it enhances, it increases the rate of this synthesis of protein. This is the first. Second, अगर हम बात कर लेंगे, it binds, second, it binds mRNA, it binds mRNA and tRNA to ribosomes. So, first इसकी बात है, it catalyzes protein synthesis. Second बात, it binds mRNA and tRNA to ribosomes. Third function, what it is going to be perform, that it provides larger surface, this area for spreading mRNA, over ribosomes during translocation process during translocation process of protein synthesis. Protein synthesis. So, what we have studied the three important functions of this. That is, it catalyzes protein synthesis. That means it enhances the rate of this synthesis of protein. Second, it binds mRNA and tRNA to ribosomes. Third, it provides large surface area. किसके लिए? For spreading mRNA over ribosomes during the process of protein synthesis. Now, these are some of the functions. Now, if we try to draw the structure of this ribosome, here again one point I will give you the term. अगर हम बात करेंगे the term, जो हम कहेंगे term, this what we call as the rRNA, it was given by term, that is rRNA. It was given by Kurland. It was given by Kurland. Regarding the structure, if I draw the structure of this rRNA, what you will see here, you will see some loops here and there. Now, what we will see, it may be the sum of, this is a three-dimensional structure rather, but it is a single stranded. This is the 5' end and this is the 3' end. So this is the single stranded DNA. What we will see, the single stranded DNA, but what we see that there are loops here and there. 1, 2, 3, there are loops here and there are formed, and these loops are formed due to the presence of what we call it, due to the presence of complementary base pairs. Bases found, complementary base pairs found in near bases. It is from base pairs नहीं, complementary bases found in near, आसपास से, in near, in the structure of rRNA. So what we will see that you will find it is a single stranded RNA, and here you will see some loops are formed, and these loops are formed due to the complementary bases which are found in the structure of rRNA. And this structure जो हमारे पास इसका है, it may be what we see, this structure, it may be either helical shape इसकी, helical, or अगर हम बात करेंगे it may be rod shaped. These are one point. And second point, अगर हम इसकी बात करेंगे, it has got a three-dimensional structure. It has got a three-dimensional structure. But अगर हम कहेंगे no detail, rather this information about the rRNA is given in the books. Thus it is somewhat what we call as the neglected topic, but not too much neglected topic, because it plays a very much important role, जो इसकी एक function है, that it catalyzes the protein synthesis. So this was all about this rRNA, and so today we have completed the different types of this rRNA. In the next video we will try to proceed further in this molecular inheritance. Until then, goodbye, God bless you, thanks.